Okay, this is Issues in Biotechnology, the way we work with life. This is the part we are discussing the applications of biotechnology, and this next series concerns pharmaceutical biotechnology. In the first part, we will discuss basically pharmaceutical biotechnology and how DNA-based technologies have led to emergent technologies today. I will cover history, alternative therapies, and aspects such as vaccine development and antibody-based drugs. In the second part, we'll focus on pharmacogenomics and the impact that genomics technologies have had on personalized medicine, the ability to use personalized genetic information and what that will bring for the future. I think this also gives us some insight into who we are and how we will then approach pharmaceutical development in the future towards personalized medicine according to our own genetics. So pharmaceutical biotechnology and emergent technologies brings up questions about how did this develop in the first place? And the problem of human suffering. The problem of human suffering has been long considered by philosophers throughout the ages, including Plato and Epicurus, Socrates, St. Augustine, through Thomas Aquinas, and more recently Albert Einstein, Bertrand Russell, Mark Twain wrote about it. John Lennon sang about it. So the problem of human suffering has been philosophically considered by great minds for a long time, and I think most of us would agree that there is still a lot of questions and unresolved issues. But certainly this is the basis for how we consider how we treat ourselves, both pharmacologically as well as through the practice of medicine. How many innocent Iraqi civilians died violent deaths during the American involvement in that country? And you can suggest your own answer, and it's somewhat staggering. These numbers were from the CRS report for Congress, reported in 2008, and they haven't been updated. But the estimates actually vary considerably depending on who is making the count and what constitutes those casualties. So you can see in this range from in 2008, 94,000 plus, to the Johns Hopkins statistical study ranging upwards or almost to 800,000. So this actually stirs many emotions and concerns. One of them would concern intervention. And I mean this in terms of the double-edged sword of intervention that conflict brought. When do we intervene? Is empathy uniquely human as an emotion, as a characteristic in humanity? Or is it present in other organisms in nature? How do we intervene would be yet another question. Let me suggest this dilemma. Imagine that you are standing on a train platform. A train is coming down the track, and you can see the conductor through the window frantically waving his arms and indicating that there are no brakes. When you look down the tracks, you see 5 men that are working where the tracks abut, and they will be crushed by the incoming train. You quickly notice that there's a lever, which if you throw it, it will divert the train to another track, saving the lives of the 5 men. However, there's a man working over there on that track who certainly will be killed. You can decide to A, throw the lever, B, not throw the lever, C, simply state, this is not my problem. D, you could stand and watch, or E, turn and walk away. Some of these are conflicting, but you can see that certainly people in our population result to these kinds of dilemmas all the time with facing these kinds of conflict. In my class, the vast majority of people would decide to throw the lever. Let's change that a minute. Same track. Again, same train, same problem. The conductor's waving his arms, there are no brakes. There are again those same 5 men at the abutment who will be killed by the incoming train. This time, however, there's no lever to divert the train. But there's a very large man standing next to you who is unaware of this situation. Perhaps he's reading a newspaper. You yourself are not large enough to stop the train if you were to jump in front of it and sacrifice yourself, but he is. If you push him or drop kick him onto the tracks, his body will stop the train, saving the 5 men once again. Of course, he would perish. So you decide to A, push him onto the tracks, B, not to push him, C, this is again not my problem, D, stand and watch, E, turn and walk away. Now when you present this to a large audience, everyone gets uncomfortable, but you can see how the numbers shift. More people would decide not to push him onto the tracks, even though the equation is the same. How do we intervene? How do we decide to intervene? Is what this dilemma exposes in all of us. And we hear in the newspaper occasionally about people who drive past accidents, people who leave hit-and-run victims in the street. There was one notable one in Hartford not too long ago that was captured on video. Before we leave this dilemma, let me pose this just one more time with another variant, and you can think of other twists on this. Same train, same situation, same problem with the man standing next to you that you could decide to push or not. This time you happen to know that he's a Nobel Prize laureate. Now, does that change your mind about whether or not you push this guy or not? Some people, this would weigh into the factor. You could also change this up, and it did, by the way, in the larger poll of people that I took about this question. Now, however, if you change that up again and you make that person a convicted pedophile, I think you can see that the majority of people result in pushing him onto the tracks. So therein is a value judgment on a human life. How do we intervene? How do we choose to intervene? Is a dilemma behind how we make medicine, how we make pharmaceutical drugs, when do we decide to treat people, who do we decide to treat? So, that's my lead into pharmaceutical biotechnology. What is it? Well, and the basic premise of this, I would like to focus primarily, as in the rest of this course, on DNA-based biotechnology. Some of this leads us into RNA-based technologies, protein-based technologies, but generally focusing around DNA-based technologies. But first, before we get to that, where do our medicines come from? What is the history of pharmacology? And this leads into alternative therapies and science. And then I will investigate how DNA-based biotechnology is used in current pharmaceutical drug development and improvement and what we can expect into the future. So first, where do our medicines come from? Drugs and medicine are intertwined. Perhaps it's an artificial construct here in this class to separate them, but we will look at this in terms of how we use chemicals in a medicinal fashion. And there is, if you look at the history of pharmacy, that peoples have been using chemicals to treat themselves in ailments for a long time. And you can look at the anthropology of medicinal use and how that varies around the planet anthropologically. Where did the early drugs come from? How did some people decide that if I eat those berries, I can cure this ailment? Did we have someone in a tribal situation say, hey, I ain't got a good idea. You go out and eat the berries. And the rest of you, if he doesn't come back, stay away from those berries, right? So there had to have been, at some point, a primitive experimental protocol. But also, it's likely that peoples observed animals and how animals actually would use certain plants, fungi, or microbes to alleviate unpleasant symptoms. And there's evidence of this still today from birds, insects, and so on. So did humans perhaps take these cues and acquire knowledge through observation and then experimentation on themselves? And this led through trial and error to the accumulation of knowledge that was passed down probably through shamans, storytellers, medicine men, and other practitioners. Certainly women in tribes in some cultures played a large role in this, but eventually accumulating this knowledge into herbal medicinal botanicals that were eventually recorded in some cultures, most notably in China. This book, the cover of which is shown in the lower left, is called Wild Health by Cindy Engels, and it describes the use of plants and microbes and fungi by animals that could have given clues to humans in various parts of the world in terms of which plants are other biological sources may have provided some medicinals. The book that's on the lower right there is called The Botany of Desire by Michael Pollan, and I, in the agricultural biotechnology sector of this course, referred to his book on the omnivore's dilemma. But this book, The Botany of Desire, examines a question that anyone who has had botany understands quite well, but it's sometimes novel to people who haven't considered this notion that some plants have done a very good job at using humans for their own reproduction. And I brought this up when we talked about corn. That certainly corn has been very successful as an organism to use humans for its own propagation. And you know, that sounds like I'm flipping things around, but look at this in terms of coevolution, that they're mutually dependent upon each other. In fact, corn wouldn't exist without humans. So in this book, The Botany of Desire, he looks at 4 crops that humans, 4 different plants that humans have used and how those plants have used humans. But in The Botany of Desire, he also examines the use of marijuana and how marijuana has, and the genetics has been influenced by humans. But he also talks in this book about how animals self-medicate for purposes of intoxication, but also for self-medication. Cindy Engels describes mammals, including gorillas, chimps, and elephants, that eat clay-rich soils that contain certain essential minerals, but also would bind toxins that might alleviate symptoms like diarrhea, which actually could result in death of those organisms. And there are people today who still practice the eating of clay, and in our country we sell a product called kaopectate that really results in the same effect. So animals not only partake in intoxicating substances, but also stimulating refreshments. Coffee is reputed to have been discovered about 1,500 years ago when a goat herder noticed that his goats became excessively energetic after feeding on red berries of a small shrub, and coffee then became cultivated for that purpose by people. And ancient Peruvian Indians observed that llamas chewed coca leaves while carrying heavy loads on a long journey and took up the habit themselves. And we can see where that has taken us. So I mentioned that humans aren't the only animals to enjoy behavioral modification, and in fact, I'll talk about this in a minute, but intoxication turns out to be on the list of human universals through modification or through ingestion of plant, fungal, or microbial compounds. And animals and birds have a strong predilection for alcohol, which they can obtain by fermented fruit, which may explain why some birds sometimes fly into my windows. I'm not sure if that's the reason, but perhaps. And the animals have been observed to overindulge, despite the associated risks from accidents like flying into my window or predation when they are alleviated of their judgment. But perhaps alcohol is consumed also because it's rich in calories and a stress reducer. So there are many examples in the animal world where animals pursue medicinals. Pharmacy, however, as a human practice, refers to the health profession that links the health sciences with the chemical sciences, and is charged with ensuring the safe and effective use of pharmaceutical drugs. So in modern pharmaceutical applications, we see that pharmacy is charged with ensuring the safe and effective use of pharmaceutical drugs. And I'll get back to this in a minute when we consider herbal medicinals, alternative therapies, and why the FDA was originated. The word pharmacy itself derives from the Greek pharmakon, which means drug or medicine. Today, this practice is pursued by pharmaceutical companies to make compounds for commercial development, and it's the sales and entrepreneurial endeavors that result in pharmaceuticals for the population. Now this brings up an interesting situation, that this is a market-driven enterprise, so that we can ponder why certain drugs are produced and why certain ailments are not pursued by pharmaceutical companies until you understand the commercial backdrop to this. There are compounds, for example, known as orphan drugs, which are not commercialized because there's not a market for it. And there are other markets which have been pursued that perhaps wouldn't affect a large global population per se, and that there are needed health concerns and attentions which could be drawn to dengue fever, malaria, HIV, which don't receive the attention, say, that cardiovascular diseases do because of the market potential and the money behind that. So here is a list of the companies that rank according to sales and where they are based and headquartered. And you can see from the headquarters of these companies that they are largely in the United States and Europe. Pfizer ranking at the top with annual sales of $43,000 million. So who regulates drug safety? If we say that pharmacy is charged with safety, who regulates that safety if there is a commercial incentive? That will bring up the origin of the FDA. The Food and Drug Administration is the oldest comprehensive consumer protection agency in the United States federal government. Its origins actually go back to a group of physicians in the early 1800s, but its actual origin as a protection agency, it can be traced back to the appointment of Louis Beck, who was in the patent office at the time around 1848, to carry out chemical analyses of agricultural products. So actually the origins of the FDA are within the Department of Agriculture, owing back to about 1862 in the United States. And it wasn't until Harvey Wiley, who was the chief chemist of the Bureau of Chemistry in the Department of Agriculture, was the driving force behind the Pure Food and Drug Act of 1906, and that's when we see the actual origin of the Food and Drug Administration. So why was the Food and Drug Administration pursued as a protection agency in the first place? Why was there a need in the U.S. government for such an agency to oversee the production of pharmaceuticals? Well, this actually owes to the rich commercial enterprise during the 1800s of various kinds of, I'll just phrase them as snake oils, that could have been attributed to anything from restoring your hair to anti-fat to making you more beautiful, making you thin, whatever it was in the bottle that was purported to do this could be sold. And it was unregulated, and people were not only getting ripped off all the time, but a lot of times people could be hurt from these kinds of treatments. So the FDA was charged with ensuring safety as well as consumer protection from these types of snake oil products. So it was actually the origins of the U.S. Biologics Control Act in 1902 that was passed to ensure purity and safety of serums, vaccines, and similar products that might be used to prevent or treat diseases in humans. Is it foolproof? No. What agency is? But I would suggest that we're better off than we were in the 1800s without it. And we can all point to flaws in the FDA. Some of them make the news, occasionally, even still today. But if you look back to, say, 1962, and the use of thalidomide as a sleeping pill, which was found to have caused birth defects in thousands of babies born in Western Europe, we can see that agencies that oversaw these kinds of compounds are not infallible. And more recently, Vioxx, produced by Merck, passed FDA phase 3 clinical trials with a low number of resulting heart attacks, which weren't noticed to be a real problem until it went commercial on a much larger scale. So we can see that similar to our inspection of food safety in agriculture, we have a similar situation here, where benefit is okay, risk is not okay. But certainly oversight is better than no oversight. So if we look at some of the significant dates in the origin of the U.S. Food and Drug Law history, as I mentioned, it was early in the 1800s, 1820, that 11 physicians met in Washington to establish the U.S. Pharmacopoeia, which was a loosely put together body of information on standard drugs that were used at the time in the United States. A lot of this was in fact almost like what we would call an herbal today. And then it was in 1883 that Wiley was the father of the Pure Food and Drugs Act that was instituted in 1902 as the Biologics Control Act. But some other notables here. 1938, the Federal Food, Drug, and Cosmetic Act to regulate safety of drugs and cosmetics was introduced, and in 1939 the first food standards issued. Later, in 1958, the Food Additives Amendment required new food additives safety, and I already mentioned thalidomide and what that raised our consciousness about in terms of drug safety. In 1968, the United States FDA introduced the FDA Bureau of Drug Abuse Control Act. And in 1970, the Comprehensive Drug Abuse Prevention and Control Act replaced previous laws and categorized drugs based on abuse and addiction potential compared with their therapeutic value, and this has retained some of the conflicting information that we still have today on that issue. 1995, the FDA declared cigarettes to be a drug delivery device, and this really set in motion a lot of anti-advertising campaigns that we have seen since, and the lowering of resulting cancer rates. 2005, the Food Allergy Labeling and Consumer Protection Act required the labeling of any food that, on account of the vast majority of food allergies, peanuts, soybeans, cow's milk, eggs, fish, crustaceans, tree nuts, and wheat, might be overseen. And in 2009, President Obama signed the Family Smoking Prevention and Tobacco Control Act into law. This leads me to alternative therapies, since there are many alternative therapies that are derived from herbal medicines today that are not regulated by the FDA. These can be sold over the counter by really anyone who chooses to open up such an enterprise. Now, there's some interesting information that we have to consider about this, and I'll get back to this later, but many of our pharmaceuticals actually are derived from plants, fungi and microbes, but particularly land plants, owing to the early experimentation I described amongst early peoples. Aspirin, for example, is salicylic acid. Salicylic acid is common in many plants. It's a plant hormone, in fact, and it concentrates in black willow bark, which was used by Native Americans to treat toothache, and tea concoction was used to alleviate childbirth symptoms. So certainly aspirin was derived from a natural product in willow bark. But notice that we don't consume willow bark when we take an aspirin. We use the purified compound most of the time. I will get back to this in a minute when we consider other medicinals that are derived from plants. But one of the benefits of doing that is, first of all, a controlled dosage. Second of all, a safe, reliable resource. And third, you should consider all of the other compounds that are present in willow bark that might affect you, other than just the salicylic acid. And there's a certain reductionist thinking that goes on here. To say that willow bark is good for your toothache is one thing, but you must consider all of the other compounds and their resultant side effects that may come with that. Another thing is that the willow bark did not make salicylic acid to treat your toothache. And there's a certain anthropocentric thinking that goes on about this, resulting in incorrect thoughts about plants that produce these compounds. And there are certainly bioactive compounds in plants all over the place. They do not make these compounds, however, for human consumption. That's ridiculous thinking, really. So a lot of these alternative therapies, you can go into these shops and you see things like ginkgo biloba tree leaves will increase your memory. At least I think. I can't remember. I didn't have my ginkgo biloba yet today, I guess. But we should consider what's in ginkgo biloba. Ginkgo biloba is a conifer, actually, a gymnosperm with broad leaves. It's used as a horticultural plant these days in North America. But I'm not aware of any peer-reviewed, evidence-based research which indicates that ginkgo biloba actually can have an effect on your memory. I am aware, however, that as a plant, it has very few plant pests, and it has a large concentration of certain phenolic compounds which may cause liver toxicity. So ingesting a large amount of ginkgo biloba may have adverse effects. And this can be purchased over the counter, non-FDA regulated, and it almost appears as though we go back in time to the 1800s. And so we see a similar sort of approach in many different alternative therapies. I am not here to write these off by any way, shape, or form. Some of these therapies I think can be beneficial to people in many different circumstances. What I am encouraging, however, is an evidence-based approach and accountability on the behalf of their practitioners, and somewhat of a warning to the consumer about some of these therapies, including herbal medicine, homeopathy, chiropractic. DNA activation is an interesting one on this list. There are advertisements stating that you can activate your DNA. And I think if you read over some of these advertisements, you can see contradictions blatantly to what we understand about the molecular structure of DNA, and that for some price, you can have your DNA activated. I don't know. But I think what I'm encouraging here is an evidence-based approach, looking at knowledge and evidence that is accrued to peer-reviewed science. When we look at this in 2011, 60 million Americans had used one or more complementary or alternative therapies to address health issues. So certainly that's a large part of the population. And the research found that those having used alternative medicines tended to have higher education or report poorer health status. I'll get back to this in a minute, and the seeming contradiction of that. The majority of alternative medicine users appear to be doing so largely because they find that these health care alternatives seem to be more congruent with their own values, beliefs, and philosophical orientations towards health and life. A majority of alternative medicine use was in conjunction with standard use, and only about 4.4% of those studies used alternative medicine strictly as a replacement for conventional medicine. And popularity overall, in spite of what I said about higher education and poorer health, popularity is shown to be linked to a low level of scientific literacy or trust in science in general, and certainly trust in large pharmaceutical companies among the public at large. Many of today's well-accepted drugs, as I've mentioned, have been derived from plants. Traditional herbs, you can look at Chinese herbals, which have credence. I mentioned the aspect of aspirin. You can look to also the recent use of Artemisia to control malaria, and how that compound now has been isolated and even synthesized and has been shown to be very therapeutically effective in treating even late-stage malaria. Or look to the drug Taxol, which is now commonly used as a chemotherapy for breast cancer treatment in conjunction with a pharmaceutical drug called Herceptin that I'll mention in a little while, that is the outcome of recombinant DNA technologies. But Taxol is originally a compound found in a conifer yew plant, which has been recognized and synthesized out of the yew. So certainly there are examples all over the place where traditional herbs support this. And you can find confounding evidence as well for things that simply are nothing more than a placebo effect. And in fact, in China, the placebo effect, so-called, is higher than it is in the United States. In other words, if you believe that this will work, there is a tendency that this will work. And in fact, and we don't understand the placebo effect. What is the molecular basis for the placebo effect, so-called, is not certainly well understood, but there is a known and measurable placebo effect in many drugs, and this is often used in double-blind studies to evaluate the efficacy of drugs. So amongst all of these products, some work, some don't. Some we have evidence for, and others there aren't any. So most alternative therapies, however, are not regulated. How can we determine if alternative therapies work? Evidence-based knowledge. Take a scientific approach. If herbal medicines and alternative therapies are a multi-million, if not multi-billion-dollar enterprise, then the onus should be on them to provide their own proof or support research that would do so. This one I took from the book What is Life by Jay Phelan, looking at the scientific method, and I'll just suggest this here as an evidence-based approach to evaluating alternative therapies, or for that matter, any hypothesis. Step one, make observations. Would this berry used by other animals be useful for treating a human disease? That would be forming a hypothesis. Step three, devise a testable prediction. I'm going to give some of you this, and some of you a placebo, and some of you not anything. Conduct a critical experiment to evaluate that and draw conclusions, and then make revisions. If it's repeatable, I think you might have something. Let's look at the example in this text about people taking echinacea, which is a common herbal treatment that is purported to boost the immune system, that consuming echinacea extract may reduce the intensity or duration of symptoms of the common cold. That's an observation made by many people who ingest and ascribe to the use of echinacea. So you can formulate a hypothesis. Echinacea reduces the duration and severity of a common cold. And then you can devise a testable hypothesis. We will treat individuals with echinacea and individuals not treated with echinacea, and predict whether or not echinacea reduces the duration or severity of the symptoms. Then we would conduct a critical experiment with 4 groups shown here, comprising the experiment, so that group 1 would receive treatment, group 2, and group 1 would receive the treatment before exposure. Group 1 would receive the treatment, group 2 would receive the treatment before exposure, group 3 and 4 would receive no treatment. And you can go through these results, and researchers randomly divided 437 volunteers into such an experiment. And I think this was conducted by Johns Hopkins, but I don't have a reference for that in this lecture. So groups 1 and 2 received tablets for 7 days longer than groups 3 and 4 in order to determine if the treatment prior to exposure has any effect on the development or the duration of the symptoms caused by the common cold. So after 1 week, all individuals are exposed to a cold-causing virus. And then health evaluations for 5 days, and doctors monitored all groups according to the symptoms. Notice that group 3 received a placebo tablet daily, unbeknownst to them. And we could get into the ethics of placebos in double-blind studies such as this. Step 5, we make our conclusions, and in this data we see that echinacea had no effect on the duration or severity of the common cold. So, in fact, this data is published in peer-reviewed journals and has been repeated and held up. So the experimental conclusions often generate ideas for further experimentation, and that is the process of science. So alternative therapies then should be evaluated, I think, under a similar sort of regime. Does it work? Does it not work? What's the evidence for it? What are the possible side effects? What are the risks? What are the benefits? Seek a balanced view. You yourself would be an experiment of a single data point. Is that sufficient? If you think it is, that's quite a risk. On the other hand, if the risk is worth the benefit that you perceive, go for it, right? There are good reasons, however, to use purified medicines. We don't take willow bark anymore to treat toothache. And we see that life expectancy is up worldwide in the past 100 years. And this is not merely a coincidence. So alternative therapies, all of those on that list, I think should be subjected to scientific scrutiny. Efficacy, do they work? On what group of people? And avoid the tendency for reductionist thinking. This herb is for that disease, or this herb was made for that disease, is even worse. The absence of science-based results should raise the risk factor, and perhaps we should require labeling to draw attention to the wary consumer. In the absence of regulation, there should be accountability. Right now, there is no accountability. If I sell you a bag of hay in the alternative store down the road, I'm not accountable whether that worked or not, for one thing, or whether that had deleterious side effects. So I guess my cautionary tale here is that there is a need in this industry for evidence-based studies. Broadly, I would say there's a need because the consumer, 60 million of them in the United States, are drawn to these kinds of therapies. And there's a need also for education on this topic. And I really don't take this lightly. And the reason I don't take this lightly is alternative therapies can kill you. And either directly or indirectly. And I say indirectly in this case because a friend of mine had breast cancer and chose to treat this with unfounded therapies, and well, I think the result would have been the same untreated. That would have been my assessment there. Okay, so that's history. Where does this lead us now? How will DNA-based biotechnologies be used in current pharmaceutical drug development? In the past, we have seen this progression from herbal medicines to from drugs that have been extracted out of plants and fungi into now our current era of innovative thinking and engineering intelligently about these molecules that we can use to treat diseases. I am sure that half of you would not be here without antibiotics. And why am I sure about that? The demographics say so. How many of you have taken antibiotics? How many of you wouldn't be here hadn't you? And we can almost not even imagine a world before penicillin. Similarly, we can almost not imagine a world without anesthetics. Do you realize that we went through the Civil War without anesthetics? That's hard to believe. Let alone antibiotics that could have killed the disease, antibiotics that could have killed infection that certainly resulted in a lot of deaths otherwise. So where does this lead us today? I first want to look at small molecule drug design, because a lot of the pharmaceuticals that we have used and developed over time, even derived from plant material, are in fact small molecules. Then I want to talk about vaccine development and some of the myths and facts concerning vaccines, how they're developed, and antibodies. And then we will eventually get to recombinant DNA drugs. So first, small molecule design. If we look at the history of the pharmaceutical pipeline in this slide, the first generation refers to what I just discussed about using natural products and their derivatives. And basically this relied highly on serendipity. Finding penicillin, for example, was the result of a contaminated Petri plate. And other of these, I don't mean it was strictly dumb luck, because that observation was keen and it was acted upon. But now into the second generation of drug design, looking at the development, say, of NSAIDs and their effect on certain receptors and enzymes and targeting antagonistics, and things like beta blockers came to be in the 1980s. And then eventually, starting actually in the 1970s, the use of genetic engineering and using clone DNA to make drugs and evaluate protein-based drugs. The third generation I'll talk about later on in this series, using DNA-based knowledge, that is genomics, using DNA sequences and the knowledge that that brings for personalized drug development and drug design. So first, small molecule drug design. Many of the drugs that we now have for various diseases are in fact small molecules and chemicals. And in the pharmaceutical industry, designing small molecules to fit specific sites, whether it's to fit certain protein sites or to fit uniquely into DNA itself, is one of the aspects of that type of research. So designing certain molecules that will uniquely fit into a receptor site as a protein and either block it or activate it is part of the design feature that people are looking at. Rather than randomly selecting bioactive compounds from plants and seeing what works, actually designing a small molecule to fit its biological receptor. And the use of other tools like combinatorial chemistry, also known as test tube evolution or accelerated evolution, being able to tweak molecules in all of their different conformations and test them against their ability to fit certain receptors and then further tweak them. So now using arrays such as we see in combinatorial chemistry applications has resulted in a number of efficacious new drugs. So those types of tools come to bear on small molecule design within pharmaceutical companies today. And there is also the ability to examine activities of chiral drugs. What is a chiral drug? Well, in biology in general, chemistries come in D and L forms. These are isomers of each other that are mirror images, like your hand. So you would have right and left-handedness. There are D and L forms of amino acids, for example. So notice that these are mirror images. They're not superimposable, but they are in fact direct mirror images like your hand. It turns out that biological activity of a D form may have different or even better activity in its L form. And so the investigation of chiral activities in small molecules has also resulted in new and better drugs. And certainly these tools, like investigations of chiral chemistry or combinatorial chemistries, has vastly decreased the time of new drug discovery and development, but there is still a huge pipeline of molecules that is examined to come down to eventually the one or two that drip out the pipe at the end. And most drug companies have vast libraries of these things which they evaluate first for their biological activity, next for their safety issues, and then for phase 1, phase 2, phase 3 clinical trials, spending vast amounts of money. And this has really gotten to be a lot more intelligent than it was, say, in the 1950s, which was almost a spray and pray approach. You know, looking at molecules that had biological activity and then finding out whether they had specific activities against a certain disease, and then whether or not they were safe on a large scale. So modern tools of chemistry have decreased that spray and pray approach down to a more focused idea about biological activities on small molecule design. And I'll talk later about how the ability to invent and synthesize new small molecules that are effective to binding certain receptors comes to bear in the genomic era. Certainly, small molecule drug design is going to be affected by the role of pharmacogenomics. And I will get more into the topic of pharmacogenomics in the second part, but I just wanted to introduce it here in terms of its role in small molecule drug design. So what is pharmacogenomics? Pharmacogenomics is looking at the whole DNA sequence of an organism, and in this case humans, and it's the branch of pharmacology which deals with the influence of genetic variation on drug response in patients, correlating gene expression or single base pair changes with a drug's efficacy or toxicity. So in fact, a drug that could be good for you might not do anything for him. And it would be good to know that ahead of time. This previously has been inaccessible and unresolvable in a population until this went out on a larger scale. We now have access to genomic information which may give us clues to that. Also, owing to genomic information, we might be able to tell whether a drug that might be good for you could kill him. And that would be good to know certainly ahead of time. So the introduction of information-based sciences like pharmacogenomics will have a large influence on drug design and drug toxicity. For instance, when we now give a molecule to you and during drug metabolism it separates into two compounds, what are the effects of those two separate compounds? This previously is very difficult to get a handle on. And we can talk about side effects of this and side effects of chemotherapies and so on, but we can see now that this kind of information will give us an avenue to pursue this at a scientific and evidence-based approach. So the role of pharmacogenomics then will allow for personalized medicine treating various diseases. So I mentioned that a drug that might be good for you would kill him, but that might depend on your personal genetics as well. And personalized medicine in terms of different types of medications. We now see people on not just one medication at a time, but several. How are those interacting with your own genetics? So based on a patient's DNA sequence, we might be able to come up with a higher efficacy rate. So in the sequencing analysis of the human genome's 6 billion base pairs is now becoming cheaper and cheaper owing to better technologies. In your lifetime, you will have access to your own genetic information at this level. How that's handled will continuously be more resolvable. So I mentioned the ability to take that information and evaluate drug metabolism certainly will be an outcome of that type of information, but so will disease predisposition. And this will offer both good information, but it's a two-edged sword that is difficult to comprehend as well. Do you really want to know ahead of time if you have Huntington's disease, which is right now untreatable and results in certain death? Is that something you want to know? For one thing, it might alter the way you run your life, I don't know, but it could have devastating consequences as well. Or do you really want to know if you have a 30% chance of getting some type of cancer, treatable or not? That's a 30% chance. In some cases, that may affect how you run your life. If it's skin cancer, you might want to stay out of the sun, for example. Other types of cancer, it might not have any effect on your lifestyle, and you're just worried about it and causing you a lot of stress. So disease diagnosis, disease predispositions, all of these will fall under the rubric of knowing your own DNA. Genes and behavior. We are coming to understand that genetics do play a large role in human behavior and animal behavior as well. There is a list you can get that's in the back of a book published by Steven Pinker called The Blank Slate, which you may recognize that phrase from John Locke's hypothesis that we are born a blank slate and then enculturated. Well, this is a very interesting book because what he looks at is the influence not only of enculturation, but the influence of our genetics on cultural and social behaviors. And in the back of this book, he publishes the list of so-called Brown's human universals. And this is a list of human characteristics that have been observed widely in anthropology by various different sources in order to get on this list. And so these are traits that are across culture and across time that have been widely observed in humans. In other words, we can only say that this must have a strong genetic predisposition to get on this list. I mean, if people from Africa to South America to Iceland and Tierra del Fuego all have these same traits, you might presume that they are not the consequence of enculturation onto a blank slate, yes? So the genetic basis of behavior is certainly supporting this hypothesis in order to, and I encourage you to look at this list, because one thing that strikes me as really interesting is that it's not short. I can't remember the exact number of traits on that list, but it's long, and it's continuously being added to. But it's also interesting what is on the list, and actually what's not on the list. Some things as, seemingly benign as hairstyles. You might notice I cut my hair just recently, and all humans have hairstyles. There's not apparently a human culture anywhere that just lets it all go. They do something with it. Even Aboriginals do something with it. So it's interesting that that's on the list of Brown universals, and there's a lot of these things. But there are other ones that make a lot of sense in terms of social and moral norms that we observe around the planet. Like pretty much all cultures say it's that murder is a bad thing, and so you can see commonalities between these types of things, but it certainly underscores the idea that there are behaviors that have a deep genetic basis. And this also figures in now to pharmacological approaches to treating behavioral disorders, and we see this is becoming more and more prevalent in our society today. So there's molecular evidence now for behavioral diseases such as bipolar disorder, schizophrenia, major depression, and so on. Studies that I have seen indicate that major depression and schizophrenia may be over 80% genetic component and a 20% environmental component. So drugs can be devised that will mitigate or modify some of these behaviors, and we see these now in use more prevalently in our society today to treat or alleviate all kinds of these things. And for example, SSRIs and Prozac. Now I want to give you a story about not only placebo effect here, but before we leave herbal medicine entirely, you may have heard that drinking tea of St. John's wort is reported to alleviate effects of minor depression. And I am aware of a couple of studies that have evaluated, like my study that I showed you on echinacea, the effects of St. John's wort. Because when St. John's wort was first purported to alleviate minor depression, double-blind studies were conducted. And in fact, unlike my echinacea story, it turns out that in many cases, St. John's wort tea can alleviate some people from symptoms of minor depression. Not major depression, but minor depression. So drug companies were fast onto this. Is there a compound in St. John's wort that would be Prozacian? Is that a word? Or Zoloftian or something, you know? Was there a SSRI, a serotonin reuptake inhibitor, that had an effect at the molecular level of neurotransmission in St. John's wort that could be synthesized out the way we recovered aspirin? So people started looking for compounds in St. John's wort that would have such an effect, in that they had evidence that in fact it does alleviate minor depression. And they did these studies on neurotransmitters and over again could not find a compound in St. John's wort that affected neurotransmitters. So running into all of these hard dead ends, eventually it was realized that St. John's wort increases cytochrome P450s in liver cells. And the effect of this is that this increases the detoxification capacity of liver, increasing detoxification enzymes in the liver. And it was the fact of alleviating mild liver toxicity, and it was liver toxicity that was resulting in the minor depression. So rather than affecting neurotransmitters, it was the alleviation of mild toxicity by increasing cytochrome P450 and other liver enzymes that resulted in the alleviation of those apparent symptoms. Oh, I should remind you that this may also have an effect on some drugs of decreasing their effectivity. So if you're taking a medication for something else and you increase your liver detoxification enzymes, it may result in the decrease of the effectivity of certain medications. So perhaps St. John's wort should come with such a warning label. For example, the contraceptive pill, for example, might have a decreased effect if you're drinking St. John's wort tea in that. You might want to know that. So like if you think you're depressed now. Anyway, drugs and behavior have been well correlated over time, and I brought up the Food and Drug Act on drug abuse when I gave this history, because our society has interesting ways of evaluating illegal or socially acceptable drugs from those that are legal and prescribable, as well as foods and supplements. It could be said that some compounds that we ingest routinely as food should be treated as drugs. In fact, some people are making this claim for all kinds of common things that we don't normally consider to be drugs. Sugar is a drug. Look at the effect it has on little kids. Is that, you know, the little kid that was just here, for example, you know, it's like sugar is a drug and not commonly attainable in wild environments unless it is synthesized out of products like sugarcane or other sugar-bearing compounds. Chocolate has an effect. Tea, alcohol, certainly. Other supplements that we consider to be legal drugs. And then on the other side of the equation, there are drugs of abuse which tend to trigger reward response neurotransmission, and that then accentuates their addictive tendencies. Some, not all of the drugs shown here of abuse are addictive in the strict sense of the word, but certainly these are handled with care. On the other hand, there are some drugs which have been deemed in our society as illegal. And some of these are merely plant products, but because of their use or perceived abuse in our society, or for one reason or another, have been deemed taboo. And some of you may be old enough to remember the campaign that started the so-called war on drugs in the United States with the cracking of the egg into a sizzling frying pan, and this is your brain on drugs. I don't know who came up with this ad, but it stuck with people for a long time. You're laughing, I can tell, that you've probably seen this ad, but this has nothing to do with neurotransmitters and how they actually behave in the presence of any of these compounds. Let me ask you something. If you knew your genome and it revealed that you had a genetic recessive trait for schizophrenia, you yourself then would not have the disease, and your spouse had the same diagnosis, would you want to have a prenatal screen showing that your child-to-be may have the predisposition for schizophrenia? And if you did, then what? Okay, what are the emerging technologies then that will impact pharmaceutical research and development? I should, before I get to that, demonstrate the commercial impact of these things. I mentioned orphan drugs, that we treat some diseases and other ones, even though we have drugs to treat them, are not marketed, simply because there isn't a large enough population. Now, what do I mean by that? Well, a drug right now to get to market is going to take 13 to 15 years and well over $1 billion in order to make it into market. So if you've got a disease that only affects, say, 50,000 people, you are not going to recoup that investment, even though you have a drug in the lab that you know will have an effect on 50,000 people and even save 50,000 lives. There's not enough money in order to encourage the development of that drug. On the other hand, you could have a drug that, say, will treat erectile dysfunction. It's not killing anybody, but you're going to get a lot of money out of that. Therefore, that goes through. People are dying of malaria, but there is Viagra. Interesting. People aren't dying of malaria in this country, but the people that are dying of malaria can't afford to pay for this kind of a price tag. HIV similarly. If you saw a large population of the United States affected by malaria, I'll bet you anything you would see a malarial drug on the market pretty quick. Same thing with HIV. And the road to a new medicine is actually quite long, and there are a lot of pitfalls. So the pharmaceutical companies actually enter into a large risk situation investment-wise. This is why they're large. This is why drugs are expensive. The failure rate is high all along the road, from the light bulb going off from the idea of the scientist in the lab, through drug discovery, exploratory development, full development, phase 1, phase 2, phase 3 clinical trials, full development, and then post-commercial analysis, such as now what we receive in the aftermath of the Vioxx experience. So an attrition to this R&D process is long. resulting in only 1 or 2 drugs that actually make it out. And think about the effort that goes in to some kind of compound that fails phase 3. What a bummer. You went through all of this and all of the expense to have something that fails at the finish line. Sort of like, what was that horse that lost in or didn't even get to compete in Belmont recently? You know, kind of like that. Yeah. In spite of that, the percentage of sales that is contributed to R&D in the pharmaceutical industry is quite high, and it needs to be in comparison to other industries noted on this list. Look, for example, at software and media services, which obviously is a lucrative kind of an enterprise. Most of you are sitting there with one in your lap or at least one in your pocket, or maybe a couple. But the sector does less than a half, I think, of investment in R&D to what pharmaceutical companies put into drug development.